Hi, everybody. So I was asked by a student to do some review on the cytoskeleton, and I decided to do that by a video because I can do so much more in that limited amount of time than I can write in an email. And while I'm making a video on the cytoskeleton, I figured I would just make it for all students and share it on Kodiak. So what you're looking at, hopefully, if this is working well, is basically a whiteboard, and I'm just going to doodle on the whiteboard and explain the components of the cytoskeleton as best I can as I do so. Be focusing on the structure of the cytoskeletal components as well as their different functions in the cell and give you a good sense of what you need to know for Monday's exam. So the first thing is, what are the different components of the cytoskeleton? And we have microtubules. That's not going to work. So I'm just going to abbreviate them. We've got microtubules, or MTs. We've got intermediate filaments, or IFs, and we've got actin filaments, or microfilaments. I'll always refer to those as actin filaments. And I've written these in descending order of thickness. So microtubules are the thickest. They're actually hollow. Intermediate filaments are intermediate in their thickness, and active filaments are the least thick. They're the thinnest of the three. So structurally, I guess we'll talk about the microtubules first, since I listed them first. Microtubules are made up of individual monomer proteins, individual subunit proteins linked end to end. And those subunit proteins create a chain of tubulin. Tubulin is the name of the individual proteins. They actually come in two different types, alpha tubulin and beta tubulin, but I won't ask you that for the exam. So we've got these tubulin proteins that are roughly spherical in shape. We refer to that as globular. And we make one chain, and then we do that again, and we can make another chain of tubulin, and we do that again to make a third chain of tubulin. And we keep doing this, and we stack these tubulin chains side by side until we make a hollow tube of tubulin. So what you're looking at here is a microtubule from the side, as though we're looking at it from this orientation. But if we look at a microtubule down its end, this way, down its front end, we would see each of the terminal tubulin subunits making a ring like that, so that we're looking down the caliber of the hollow microtubule. It's actually 13 different chains of tubulin that make a microtubule. I'm not sure if I drew 13 circles here in this view, but that's the overall structure of a microtubule. So they're pretty fat. They're made up of these globular or spherical protein subunits, multiple chains stacked together to make a hollow tube. Second are the intermediate filaments. The intermediate filaments couldn't be any more different than the others, because the individual subunits of intermediate filaments, the proteins themselves are quite linear. They're not globular, they're not spherical. The proteins are linear. And so typically what we have is a tetramer of the intermediate filaments coming together to kind of make a braided rope or some kind of tangle. And then we take tetramers and we braid them together. And we continue doing this until we make very long chains of intermediate filaments. But the intermediate filaments, there's nothing globular or spherical about them, even in their subunits. They're very, very fibrous, very linear. And this is why intermediate filaments are the cell's rope. They're ideal for tensile strength. Intermediate filaments can stretch without tearing or stretch without breaking. And so intermediate filaments basically hold the cell together and allow the cell to stretch without tearing. And our tissues as well can stretch without tearing because of intermediate filaments. And then the last are the actin filaments. The actin filaments, very crudely, resemble microtubules more than they do intermediate filaments, because the subunits of actin filaments, which is actin, are these more globular spherical proteins. We make one chain of actin monomers, and then we make a second chain of actin monomers. And as I'm drawing it here, we take those two individual chains of actin monomers and we basically twist them together like two twist ties. So it's not nearly as durable as intermediate filaments, not nearly as thick as microtubules, but have some elements of both. Actin filaments are similar to microtubules because the subunits are more globular, and they're similar to intermediate filaments because they tend to be more like chain-like, and they're not hollow on the inside. They're not tubes. So we talked a little bit about the three different types themselves, their names: microtubules, intermediate filaments, and actin filaments. And this is, in general, how they're made, or at least how much you need to know about how they're made for this exam. So the last thing we'll talk about is their function. And for that, I want to put us in the context of a cell. So here's the cell membrane of a typical eukaryotic cell. Put the nucleus here, put the nucleus in as a double membrane to try to make it as accurate as possible. And we'll leave it at that for now. So usually right next to the nucleus, pretty close to the center, is at least one centrosome. And coming out of that centrosome is, let me see if I can find thick, here we go, is or are the microtubules. So the microtubules emanate out from the centrosome, and they, just as I'm drawing them here, they permeate and go through the entire cell, making up this network of highways and byways of the cell. Next, we have the actin filaments. The actin filaments, I think I drew them as black as well. I'll leave them as black for this, I guess. The actin filaments, in the context of cell structure, create the cell cortex. The cell cortex is this meshwork of proteins that lies just beneath the cell membrane. It almost acts as a protein cage or sphere, and the cell membrane is draped over that cell cortex. That cell cortex is made up of actin. So I know I said this in class plenty of times, but keep in mind that the consistency of the cell membrane is equal to about olive oil. So the cell membrane itself has no structural stability at all. It will just kind of mush out if it doesn't have some kind of structural support under it. And the structural support of the cell membrane are the actin filaments of the cell cortex. And finally, the intermediate filaments, well, they can just take up the rest. I mean, I could fill this whole cell with just a vast network or mesh of intermediate filaments. And the intermediate filaments are going to give the cell its three-dimensional structure, its stability, and provide that tensile strength for the cell, allow the cell to be somewhat deformed and somewhat stretched and put under strain without tearing. So the intermediate filaments are doing that. Also, the specific form of the intermediate filaments called the nuclear lamina or the nuclear lamins, they create a protein cage or a protein sphere under the nuclear membrane. So what the actin filaments are doing for the cell membrane, intermediate filaments are doing for the nuclear membrane. And also, we should just point out kind of on the side, keratin is also an intermediate filament. Keratin is the protein that your hair and your fingernails are made out of, so that's intermediate filament as well. So that's kind of each of these structures or each of these cytoskeletal components in the context of the cell. The last thing we'll talk about, I guess, is the motor proteins and what kind of movement is done with these cytoskeletal components. So for the intermediate filaments, it's easy. None. There are no motor proteins that move on intermediate filaments or pull intermediate filaments. Intermediate filaments are static. They're purely structural. However, for actin and for the microtubules, we do have motor proteins. For the microtubules, those motor proteins are dynein and kinesin, those walking proteins. And the only thing different about them is the direction that they walk in, which we really don't have to concern ourselves with. But both of those proteins, dynein and kinesin, carry vesicles, vesicles that are loaded with proteins, proteins being shipped from the rough ER to the Golgi, from the Golgi to either organelles or the cell membrane, or organelles that are coming in from endocytosis, things that the cell has brought into itself, and maybe it's shipping that material that it just brought into the cell in a vesicle to the lysosome for digestion. So vesicles are being moved throughout the cell by motor proteins, dynein and kinesin, but those motor proteins are walking along the microtubule tracts of the cell. And that's why I always refer to the microtubules as the highways of the cell, because the motor proteins, which are carrying the cargo, are moving on those highways of microtubules. Microtubules also, incidentally, pull newly replicated chromosomes apart during cell division in a process called mitosis that we'll talk about a little bit later on in the semester. But the cables, the protein cables that pull newly replicated, newly copied chromosomes apart are also microtubules, but that only happens as a cell is dividing. The last kind of cellular motion that I'm not sure how well I can draw on this, but I can do my best, is when the cells themselves are moving. So cells themselves will move. In our own bodies, white blood cells are motile when they're fighting infection. A lot of our cells move around during development. Neurons grow out what's called a growth cone, and they move in a certain direction of other neurons to make connections during development. And then unicellular organisms, such as amoeba, are also on the move. And anytime cells are moving, it's most often and largely done by actin. Even our muscle fibers, where we have movement of the multicellular organism, is often actin-driven. So the motor protein that interacts with actin is called myosin. Looking for a color I haven't used yet. I think it's only this kind of pukey yellow-orange. So we've got myosin. Myosin is the motor protein that interacts with actin. And just like kinesin and dynein use energy to walk along microtubules, myosin uses energy to quote unquote walk along actin, but not quite the same way. Whenever myosin is interacting with actin, it's the actin that moves. The myosin is the lever arm, the myosin is the puller, and the myosin is pulling on actin. So some examples where we see that come into play is when a cell like an amoeba is going to crawl, actin will start pulling on and pushing, myosin will start pulling on and pushing the actin of the cell cortex, causing that actin to kind of push out the cell membrane. The cell cortex is modified and used as a battering ram, and that battering ram deforms the cell membrane. So it's myosin pulling on actin that changes the shape of the cell so that the cell reaches out in the forward direction. And then when it's time for the entire cell to kind of heave itself forward and take the place of that new location where we've just reached out to, it's other myosin proteins that are pulling on the actin of the cell cortex to heave the body of that cell forward in that direction. Also, it's actin and myosin. Myosin is the motor protein that's pulling on the actin filaments of muscle fibers, causing muscles to contract. We talked about quite a lot of that in class. So in general, right, you never want to say always in biology, but in general, when a cell is in motion, when the cell itself is in motion, actin and myosin are involved. When there's movement within the cell, when there are things moving in the cell, usually that's the interaction of dynein, kinesin, and microtubules. And then intermediate filaments are not involved in motion at all because they're purely structural. So yeah, that's pretty much what you would want to know for the cytoskeletal components for the exam from a storytelling point of view. The big takeaways are motor proteins are not part of the cytoskeleton. There are only 3 components of the cytoskeleton: microtubules, intermediate filaments, and actin filaments. Microtubules are involved in protein and cargo trafficking, serving as the cables that these motor proteins walk along and drag along the vesicles. And they also pull cystochromatids apart during mitosis. Actin filaments are involved in cellular motion. When cells are on the move, it's actin being pulled and deformed by myosin that's doing that. And the intermediate filaments are purely structural and there for tensile strength. I won't recap the structure, that's at the beginning of the video, but make sure you know how these proteins are made as well. And by made, I just mean their general configurations and how they're different from one another. So hopefully this helps everybody. By all means, reach out if you have any questions by email. Happy to clarify things any further if any questions come up. Have a good weekend.